Wrapping up aerobic metabolism, we have the electron transport chain and oxidative phosphorylation. So the electron transport chain and oxidative phosphorylation take place in the mitochondria, and that's why it's called the powerhouse of the cell. So we're going to learn how to describe the electron transport and the process of oxidative phosphorylation, as well as calculate the ATP from the complete oxidation of glucose. So here is a fun little summary table where in stage 3, which is the electron transport chain, each glucose molecule that completes glycolysis from one glucose, in glycolysis we produce 2 ATP net, okay? And then the citric acid cycle, we have 2, and then for one glucose, that means that we have 4 ATP so far, 10 NADHs and 2 FADH2s. An electron transport chain, this is also called the respiratory chain, hydrogen ions and electrons from NADH and FADH2 are passed from one electron carrier to the next, and they combine with oxygen to make H2O. The energy released during electron transport is used to synthesize or make ATP from the constituents ADP and PI, and this is a process that's called oxidative phosphorylation. Sometimes I'll call it OXPHOS, right? As long as oxygen is available for the mitochondria, electron transport and oxidative phosphorylations function to produce the most ATP in the cell. So this is where we really get the bang for the buck. So the mitochondria is where this is all happening. So the mitochondria contains an outer membrane, an inner membrane space, and a highly folded inner membrane that surrounds the matrix. And the enzymes and the electron carriers along the inner membrane are required for this transport of electron. And so 4 distinct proton complexes are there. They're complex 1, 2, 3, and 4, and these are located within these membranes. So 2 oxygen carriers, we need helpers, right? And that's what these guys are, 2 electron carriers, coenzyme Q and cytochrome C. So you can see that there's here coenzymes Q and cytochrome C are these 2 little purple blobs. So look in your book and it'll be bigger, okay? But the 2 electron carriers, coenzyme Q and cytochrome C, are not firmly attached to the membrane. They function as mobile carriers that are going to shuttle the electrons between the proton complexes that are bound to the inner membrane space. So coenzymes NADH and FADH2 then are going to be oxidized in the enzyme complexes, and they're going to provide those electrons and hydrogen ions that are needed to make or synthesize ATP. So the first thing that we're going to talk about is going to be enzyme complex 1. Remember how NADH only had 1 hydrogen that it picked up? So you can remember it because it's 1 and it's dropping it off at complex 1. Okay, and we'll see that complex 2 is FADH2, so that's the story will come together. So NADH transfers the hydrogen ions and electrons to the enzymes and the electron carriers of complex 1, and it forms the oxidized coenzyme NAD. So remember, we're going to drop off NADH plus another hydrogen, but it's not carried from this NADH plus that Q, and then we're going to regenerate the NAD plus so it can go back upstream to the glycolysis and citric acid cycle in order to pick up more hydrogens and ship them back down. And then we're going to end up forming 2 of the QHs. So the hydrogen ions and the electrons are transferred to the mobile electron carrier coenzyme Q, CoQ, and this is going to carry the, it's going to shift, and it's like a puddle jumper, and it's going to take those electrons and it's going to shift them and copy them over to complex 2. Well, in complex 2 itself, Q also obtains hydrogen ions and electrons from FADH2. So remember, NADH only had 1 hydrogen, and it's going to drop those off at complex 1, but FADH2 had 2, and it's going to drop those off at enzyme complex 2. Okay, so we have your inputs FADH2 plus that coenzyme Q, and you're going to end up having your output be FAD again, so it can go back upstream and pick up more hydrogens, as well as coenzyme Q attached to those 2 hydrogens. Because complex 2 is at a lower energy level than complex 1, the electrons from FADH2 enter the electron transport chain at a lower energy than those that are in NADH. Well, then we have complex 3. So in complex 3, the electrons are transferred from QH2 to the iron-containing proteins that are called cytochromes, and eventually to the cytochrome C, which is a mobile electron carrier. You can see this here. Cytochrome C, and this contains iron 3 and iron 2, is reduced when it gains an electron, and it's oxidized when an electron is lost. As in a mobile carrier, cytochrome C is going to carry those electrons on to complex 4. So this is like I said, the puddle jumper, right? So then complex 4 is going to be where 4 electrons from 4 cytochrome Cs are passed to other electron carriers, and the electrons combine with hydrogen ions and oxygen to form water. So one of our final outputs of the electron transport chain is going to be water. So that's one reason why you can go and feel water in your hands, moisture is from this. So oxidative phosphorylation, this is a chemoisometric model, whatever. And this is where each complex acts as a proton pump by pushing hydrogen ions from the oxidation of NADH and FADH2 out of the matrix and into the inner membrane space. And the increase in hydrogen concentration lowers the pH, and it's going to create an H+ or an electrochemical gradient. So if we remember our pH scale that we talked about earlier, it's dependent on how many hydrogens we have, free hydrogens, right? And so if we didn't take care of those free hydrogens, or as they're shipping across the membrane, we can potentially change the pH. So in order to equalize the pH and charge between the inner membrane space and the matrix, the H+ ions are going to return to the matrix by passing through a protein complex called ATP synthase. Now, looking at our root words, we know that synthase means to make. So guess what this makes? It makes ATP. So oxphos is it's going to basically couple the energy from the electron transport to the synthesis or making of ATP from the precursor of ADP. So your input here is ADP plus inorganic phosphate plus energy. You're going to use the ATP synthase in order to make ATP. So ATP synthesis specifically, when NADH enters the electron transport chain at complex 1, the energy released from its oxidation is used to synthesize or make 3 ATPs. So you can see your NADH plus your H plus half of an oxygen plus 3 ADPs plus your 3 PIs is going to end up making 3 ATPs, regenerating the NAD plus and also producing water. FADH2 enters that complex 2, and this is at a lower energy. So therefore, we're not going to make as much ATP. Okay, and so you can see your input again here is FADH2 plus the half oxygen, ADP, PI, and we are only producing 2 ATP. So looking back and seeing ATP from glycolysis. In glycolysis, which was that first process that we talked about, the oxidation of glucose stores of energy produced 2 NADH and 2 ATP. Then glycolysis occurred in the cytoplasm, where the hydrogen ions and electrons from the NADH are transferred to compounds for a transport into the mitochondria. And the reaction for the shuttle is this. We have NADH, H+, and FAD, and this happens in the cytoplasm, and then it travels into the mitochondria, which is again where the ETC is going to happen. Therefore, the transfer of NADH in the cytoplasm to FADH2 produces 2 ATP rather than 3. So glycolysis, this is what you really need to remember. Glycolysis yields a total of 6 ATP. Okay, it's going to have 4 ATP from the 2 NADH and 2 from the direct phosphorylation. And so the calculations are there. Okay, so that is good. So under aerobic metabolism, or aerobic conditions, 2 pyruvates are going to enter into the mitochondria, and those are going to be oxidized further into 2-acetyl-CoA, CO2, and 2 NADHs. And then those 2 NADHs are going to go into the electron transfer chain, and because they each make 3 ATP, they're going to produce 6 ATP in general. So the 2 pyruvate there are going to end up making our 6 ATP. So one turn of the citric acid cycle is going to be making 3 NADH, which each makes 3 ATP, so 9 ATP. 1 FADH produces 2 ATP, so that would give you 2 ATP. 1 GTP is directly converted to ATP, so that's 1. So 1 citric acid cycle makes 12 ATP. And because each glucose provides 2-acetyl-CoA, 2 turns of the citric acid cycle is therefore going to produce 24 ATP. Okay, because remember, it split off and we had to each combine, and we have to keep track of each of them. So here is your fun little summary. Okay, and so this is a great little summary, and this class teaches that 1 glucose is going to produce 36 ATP. Now the reason I say this class is because other textbooks, some of them say 30, some of them say 32, some of them say 34, some of them say 36. It depends on a lot of different commonalities and factors. And so for this class, remember 36. Okay? So here is your overall summary of the complete oxidation of glucose to CO2, carbon dioxide that we exhale, as well as water, and it yields a total of 36 ATP. So let's do a quick little study check and see how we're doing. So to classify each as a product of the citric acid cycle or the electron transport chain, pause me and come back. CO2, the citric acid cycle. FADH2, the citric acid cycle. NAD+, remember that's not NADH, we're making NAD+, that's the electron transport chain. NADH is from the citric acid cycle, as well as glycolysis, and H2O is from the electron transport chain. All right, so indicate the ATP yield for each of these under aerobic with oxygen conditions. Pause me and come back. All right, so remember, a complete round of aerobic metabolism in this class is going to do 36 ATPs. We are going to have 2 FADH2s produce 2 ATPs, acetyl-CoA in the citric acid cycle is 12, NADH is 3, and pyruvate decarboxylation is 3. Great job, you guys.